hello everybody. So, a very good morning to all of you. So, we will be starting today on lecture 54. So, of all these non-metallic species what we are talking about. So, today we will be talking about sulfur as well as selenium. How these two elements can help us in understanding biology from their point of view. So, definitely we should know about the sulfur bearing proteins, we all know about the sulfur bearing amino acids. So, different sulfur based donors which are available, we all know that for beautiful metal ion coordination like your ferridoxin and rubidoxins. Then definitely all these Metalloenzymes have very defined sulfur coordinations from the different points, that means the preserving the coordination number. Then other part of this class basically is devoted to selenium in the biological world, and the corresponding cysteine analog of selenium is selenocysteine, how it is present in different animals including us. So, going back to the basic understanding about the sulfur, probably from your school days you are learning your sulfur, they are showing atomic number, atomic weight and its corresponding reactivity and the other patterns. But for the living cells, we also know that these are the essential component. So, during the early days of this universe, when the atmosphere is characterized by lower magnitude of redox potential, that means we do not have much amount of oxygen, photosynthesis was also not known that time. What was happening at that particular time? If you have some mineral deposit or the ore deposit of typical elemental sulfur, yellow sulfur, the sulfur powders, those are indicated for element of concern for the sulfur chemical biology. So, how we can have that sulfur, then sulfur bearing different anions and even the elemental sulfur, how it can go for producing something very interesting molecules. So, if your environment is of lower magnitude of E0 value, then the reduction of the elemental sulfur which can be available to us. So, S0 we can produce H2 minus, which is after protonation gives you H2S. So, how much we can have? So, it is only in millimolar concentration level in solution, because the solubility is very less, otherwise we will have the deposition or the crystallization. So, this chemical basically the sulfur S0 can produce different transition metal ion sulfides. All we know that if you go to excavate the earth crust with the help of a mineralogist or geologist, will get many metal ion sulfides. And in your school days or college days, what we know that the different metal ions, the cationic species can be identified nicely by knowing the color and the appearance of the different sulfides, the copper sulfides, the nickel sulfides. So, the group separation table we handled also depending upon the different sulfides. So, if we have the metal ion, so if we have the metal ion in the bivalent and the trivalent state, we can have MS, the typical sulfide, or M2S3 depending upon the two different oxidation states which are +2 as well as +3. Then if the system is available, if the biological world is smart enough, whether they can use this particular assembly or aggregate. When we talk about the iron sulfur protein, what we write iron Fe-S or Fe/S. So, these are iron sulfur proteins. So, these are not typical ferrous or ferric sulfides, but these are iron sulfur proteins, and we know that two different types of sulfurs we can have in all these electron transfer proteins. So, if we start from the reduced form, that means the S0 form and the other oxidized form, the sulfite and the sulfate, what we can have? Then bacteria are very smart enough for us for doing these chemical reactions. So, bacteria can also come into the play, giving rise to the fixation of sulfur. We know that like nitrogen cycle or the oxygen cycle or the hydrogen cycle, sulfur can have therefore the cycle which can cycle between starting from S0 and ending again with S0 by giving many important species like your sulfite and sulfate. So, sulfur ion bearing organic molecules you can have. What are those organic molecules? The simplest possible known organic molecules which will be useful for our discussion, our understanding is your amino acids. So, what are those amino acids where you have the sulfur group present? So, those whether the bacteria can produce those things and how our body can also produce those amino acids, because when those amino acids are coupled together forming your peptides, dipeptides, tripeptides, tetrapeptides and polypeptides and finally to the proteins. So, proteins will have several those sulfur groups appended on the chain, on the sheet or on the helix. Then one most important reagent, the laboratory reagent we use from our again from our school days, that H2S, we use Gibbs apparatus for the production of H2S. How we produce that also we know from the ferrous sulfide sticks and sulfuric acid. So, if you have two chambers in the Gibbs apparatus, one you filled with acid and another you filled with the solid ferrous sulfide, and continuous supply of H2S as you open the stopcock like your water tap. If you open it up, you will be able to get H2S. But nowadays for the maintenance, for not knowing and not using these things, we are slowly discarding that very simple equipment. I call it the equipment basically, it is a very useful equipment when people introduce first time, as and when required supply of H2S can be available to us. Now, if we go for the biological world, we find that this similar smell is a rotten egg smell we call, the smell of the H2S. We can also get from the rotten egg. That means, egg is a biological stuff. So, that biological material through degradation can have something where it can produce the H2S to you. So, when H2S we have, it can be oxidized again back to your elemental sulfur, which can be done by some bacteria again. So, your H2S can be substrate for your green or purple sulfur bacteria, and those bacteria can oxidize your H2S, that means the reduced form is not collected over at the top. So, it is not there. So, if you can have that particular thing, that if you can have S2 minus also. So, what we can have, the reduction as well as oxidation for all these things can happen together. So, only thing that what is that biological stuff you are using. So, bacteria what you are using. So, sulfur oxidizing bacteria can have, and we tackle with that of your S0. So, S0 we try to oxidize it, and if you say produce some sulfate via sulfide, you can fix it. But why, how and when we can have that? So, if the fully oxidized form of sulfate is available to us, we know is a very simple anion like sodium sulfate, potassium sulfate, we are using for many years. So, that sulfate one step reduction to sulfide, then finally it can definitely go through S0 and then S2 minus. So, these are the most common oxidation states of sulfide from S2 minus to S plus 6. Then whether these species can be available to us for giving the very useful amino acid cysteine or the methionine. Methionine has S-methyl function only, because the cysteine residues are available to us and we can use those cysteine residues to get not only the introduction or getting that particular very useful molecules like insulin, keratin and collagen. So, the introduction of the sulfur in very different forms is important, but if we see that the cysteine is available. So, what we know that the cysteine S minus is available. It is a very good ligand, and we all know that it can bind to Fe2 plus or Fe3 plus in your rubidoxine or ferridoxine system. So, that is why our body or our system should be well matched to produce these cysteine molecules from say sulfur 0 or the sulfate, such that it can further be utilized for your rubidoxine or ferridoxine molecule. So, likewise not only bacteria, but also worms, giant tube worms GTW also use some bacteria in their trophosome to fix the carbon, is not chromosome, is trophosome, carbon dioxide using hydrogen sulfide and oxygen or nitrate and produce sugars and amino acids. So, it is a very beautiful chemical conversion can take place and can happen by the bacteria only, such that it can produce sugar and the amino acid. So, what should be the reaction? The chemical reaction what we can think of, you take H2S, what we are taking is a very simple thing, that we are not taking H2O like water. When we use H2O for our photosynthesis, we know we will be able to produce dioxygen molecule. That is why we are surviving also, we take oxygen, plants are producing that oxygen for us through photosynthesis. But here if you have H2S and if you go for similar type of reaction, you see you will be able to produce carbohydrate or carbohydrate based some glucose molecules, then some water molecules will be freed because no water molecules are there on the left hand side, but along with that of your 18 sulfur molecules you will be able to produce. So, that is important, that elemental sulfur you can also produce by this process. Then when the bacteria is available and chemoautotrophy, the bacteria can do the process which is known as chemoautotrophy, such as purple sulfur bacteria, yellow globules of sulfur are formed and which are visible on the cytoplasm of those bacteria or the bacteria residing on some plants or somewhere. So, very beautiful powders of sulfur can be seen because they are producing continuously the elemental sulfur as this 18S. This S they are producing. So, this is a S is S0 elemental sulfur. So, PSB is abbreviated as your corresponding bacteria are part of the group of bacteria which are proteobacteria capable of doing photosynthesis type of activity like cyanobacteria. But they are anaerobic in nature, they do not require oxygen, microorophilic, then they are often found in stratified water environment because this contaminated water from the hot spring or from the industry or some stagnant water also we call is the rotten water bodies, as well as microbial mats in intertidal zones. The tides we know, the ocean tides or the sea tides we have. So, you have certain tidal intertidal zones where you can have the corresponding these bacteria are there. So, these are basically of marine origin. So, that is why the marine chemistry is also so rich and the deposition can take place below marine or the marine seashore or the ocean seashore. So, when you have the oxidizing agent or the oxidizers can use all these reduced form, not the sulphide, not the corresponding sulphate, but it can use H2S, that means H2 minus, H0 sulphide because you can go further oxidation, or thiosulphate H2O3 2 minus, and various other polythionates, that means tetrathionate. So, these are all the reduced form. So, if you have the reduced form in your hand, you can go for oxidation, that means sulfur oxidizer are there. Oxidation can take place on the sulfur atom if it is sulfur 0. So, some enzymes can come. So, sulfur oxygenase. So, is not oxidation only, is oxygenation reaction, that means you can put some oxygen or you can have some new sulfur oxygen bonds, and sulfite oxidase to oxidize sulfur to sulphate, finally we find that is to the sulphate. Similarly, some lithotrophs can even use the energy because you must have not only the chemical reaction, but we all know the free energy change which is easily accessible energy for the transformation or for the conversion or for the making of new molecule. So, we will be making new sulfur compounds which can ultimately produce sugars, a process known as chemosynthesis. Okay. So, try to know by definition only what is chemosynthesis. So, we will be producing new chemicals or new compounds through some synthetic process, but is a living synthesis. What is that? Is a biological conversion of one or more carbon containing molecules. So, if you have C1 system, that means C1 one carbon containing molecule is carbon dioxide or methane, and any other nutrients more than one or single carbon containing molecule. Organic matter we can produce using oxidation of inorganic compound, that means oxidation of H2, we know hydrogenases are there, or the oxidation of H2S. So, many useful molecules we can produce. So, what are those molecules basically? So, we go further, let us think or talk about the plants also. So, in case of plants also, the condensation of sulphate like phosphate, whether these two are not similar, is that you will have only the phosphate chains like the triphosphate chain like in ATP to AMP molecules. But sometime it can so happen that sulphate can give rise to a similar reactivity pattern, that means it can attach to the phosphate end of the ADP or AMP molecule. So, when it is attaching, sulfate with ATP to form the APS adenosine 5 prime phosphosulfate, which can further be phosphorylated to give 3 prime phosphoadenosine 5 prime phosphosulfate. Do not worry about the big name and all these things, because you already know about the ATP molecules, ADP molecules and AMP molecules. So, now instead of phosphate, we have seen also, we have studied in detail about the phosphate chemistry or the phosphorus chemical biology, how the different phosphate groups can be attached. Now, instead of phosphate, if you can have the sulfate only. So, that is the basic idea of knowing this particular thing. So, you take that ATP molecule, that means triphosphate you are taking, a biological triphosphate you are using. So, the bacteria or the plant can use that thing also and bring the sulfate and try to see some reaction. What is happening then? The inorganic diphosphate anion is leaving PPI. Pi is the monophosphate, inorganic monophosphate, PPI is your inorganic diphosphate. So, out of this triphosphate molecule system, this biological triphosphate, a diphosphate is moving out and sulfur is entering. What is the product therefore? So, we will get a phosphosulfate species, a organic or nucleobase dependent or adenosine based phosphosulfate thing. Then that can further be phosphorylated. So, how that can also be phosphorylated such that you get a molecule known as PAPS and ACP. What are these? So, PAPS, PAPS. Okay. So, PAPS is this particular molecule. So, we have taken out, basically you are having 3 phosphate rings. So, 2 of them is going out like this, then one sulfate is entering and sulfate is entering here. Then if you go for this particular reaction with another ATP molecule, so you can phosphorylate further. So, that basically gives you that particular reaction, that means corresponding one phosphoadenosine 5 prime phosphosulfate, PAPS is that particular molecule. So, this ultimately what you are getting is your PAPS molecule. So, you can have some similarity in terms of the anion, the inorganic anion or non-metallic anion what we are talking about, having some role in the biological world. So, once it is formed, the bacteria will come now, can activate the activated form of sulfate for sulfation reaction, because it is a very good reagent for sulfate transfer reaction. Like the phosphorylation reaction we have seen, you can transfer the phosphate group to some organic molecule, some alcohol, some sugar. Similarly, this particular molecule can be useful for sulfation reaction, and you see you transfer this particular sulfate group which is red in color to some organic molecule. So, you can go for that reaction as a substrate for sulfate reduction. So, that can also be a substrate for sulfate reduction, because you can tackle the sulfate and you can transfer that sulfate to other organic molecule. So, it is initially reduced to sulfide in a reaction involving thioredoxin. So, now we will bring another molecule which is thioredoxin. So, that thioredoxin, if it is coming and you are having sulfate and we are talking about attachment of sulfate to your ATP molecule, or in some other way you can go for the phosphate in some other parts also for your ATP molecule. So, is adenine, sugar and the triphosphate. So, is ATP molecule, but you can go for further phosphorylated form of phosphoadenosine phosphosulfate. So, you are going for some other part phosphate. So, you will may have, you can think of in this particular point that you can have some extra sulfate molecule attached to it, or you can have the extra phosphate. So, the molecule having some burden of sulfate or burden of phosphate. So, if we can utilize this molecule nicely, the PAPS can be utilized for the corresponding sulfation reaction as well as phosphorylation reaction. And when the sulfur is transferred and we can further reduce that sulfate to sulfide, because the sulfide is also a very good reagent, non-metallic reagent for many other type of reaction, but we bring now other enzyme or molecule for that particular purpose involving thioredoxin. So, this sulfide is subsequently reduced to sulfide reductase. So, sulfide reductase can further reduce this sulfide. A large and a complex enzyme, we are not talking all these things, but what are the systems involved over there, from NADPH to iron sulfur ferredoxin or rubidoxine molecule is available, or porphyrin cyroheme is there. So, another porphyrin based system is also available, and catalyzes the 6 electron reduction directly to H2S. So, you have the sulphite SO3 2 minus having sulfur in the oxidation state of 4 plus, that will be immediately reduced to S2 minus. So, you will have 6 electron transfer reaction for the production of not elemental sulfur, that we are not producing S0, but we are producing S2 minus. Then in plants, this APS is the substrate for reduction rather than PAPs. So, this APS basically, if we can have, and APS is basically in the different plants. So, another molecule you can have for this corresponding reduction reaction, unlike your PAPS molecule or PAPs molecule. Then once it is produced, is going through for that particular reduction process, but not we are able to trap that S0 or the elemental sulfur, but can be oxidized by purple sulfur bacteria. So, because for all these systems, what bacteria is working, what enzyme is working, you know the substrate E is coming to trap the S and you are producing P. So, most of the cases these are very simple step reaction, but we need to know the corresponding catalytic cycle if there are different steps for proton transfer, for electron transfer, and for any other shift type of 1 to shift type of thing can happen. So, then this oxidation, so reduction, oxidation, all these things are going together, as we have seen also in case of your iron sulfur proteins, because these are to and fro movement. So, whenever you have the electron demand, the system that iron sulfur proteins are getting oxidized, but when you require to put electron, it will be reduced. So, this is a to and fro reaction. So, if we go for this use of this H2S, so further oxidation of the elemental sulfur S2 minus to S0, then we bring the sulfur oxidizing bacteria, which can produce finally again back to sulphate. So, this is the window basically for the different oxidation states of sulfur S2 minus to S plus 6 in sulphate. Now, we slowly see little bit about the corresponding selenium thing. So, which is very much similar to that of our sulfur. So, sulfur we are having, we have seen that we can have the amino acids, we can have the corresponding iron sulfur proteins and many things. But what about selenium? Is the newest member in all this biological world, is the newest member in the amino acid family also. So, we should know about element wise, from the periodic table also you see how we can compare sulfur, selenium and tellurium also. So, side by side how sulfur and the selenium property and all the basic property for these, because we are having something where you are just simply instead of capital S as the elemental sulfur, you put one E only. It is not like that, you are having a difference in the elemental composition from sulfur to selenium. So, when people identified the composition of the elemental selenium, its smell is very bad, and that particular point is ugly smelling nuisance people call it, and cause chronic intoxication of farm animals like the poultry animals like the chicken, hen and all other. So, the farm animals are suffering. So, there will be toxication or intoxication by this particular element, where you can have the source of selenium as the corresponding ore or mineral, and if some bacteria is available, now it is clear to us, to everybody also, that the bacteria can do some marvel, bacteria can produce something in the elemental form, bacteria can produce the sulfur in the H0 state, the sulfur powder it can produce. Similarly, if the bacteria is available which can be active on the substrate material which is your mineral to produce the free or AC0, that means elemental selenium. But finally, we have seen that is not only very bad in terms of its order, in terms of its toxicity, but also it can be very much useful for rats and subsequently for other mammals, animals including us. So, what is that enzyme? If we think that selenium is a very important component for our survival, for our life process, so we must have some selenoenzyme. So, if we consider, okay, the statement is given to you, is reported to you that okay, we know selenoenzyme, So, definitely we must have selenium bearing protein, polypeptide or selenium bearing amino acid. So, where it has been identified. So, selenoenzymes are there for the function of glutathione peroxidase, again handling of peroxidase molecule, that means peroxidase activity. So, we have to reduce the corresponding reactive oxygen species level in our body. So, we know the peroxide concentration can be diminished, can be reduced by the catalase activity and peroxidase activity. So, similarly, if it is glutathione based, glutathione is a very useful, very simple tripeptide molecule, and that tripeptide molecule can be very useful in other purpose also, that it can show that corresponding peroxidase activity. So, 2 more Clostridia we can have, followed by a quick succession by 2 more Clostridia, a component of glycine reductase. So, one glycine reductase you must have, which is there and which is the glutathione peroxidase type of activity we can find due to the selenoenzyme, and another is your formate dehydrogenase. So, these are by name only. We are not going to study all these things, but only thing you know that where we can find this selenium. So, it can be incorporated via that amino acid to this protein molecules, you get that ACC. So, cysteine we know CYS we abbreviate, but it is SEC, is a selenocysteine residue. So, amino acid is abbreviated also in that way. So, the selenocysteine has a pK value is lower of 5.2 compared to your cysteine, which is 8.5. So, definitely the property wise everything will be different. It is a trace essential element to us, regulating the immune function, which is very important through redox regulating activity. That means, it can reduce the ROS concentration in our body and protecting our immune cells also for oxidative stress, because we are oxidizing with time. So, what is that glutathione peroxidase? Only the basic block diagram will see, the catalytic cycle will see how the glutathione peroxidase can work, no detail, nothing, do not worry for that. So, this is the thing that GPX is there, and if his GPX is cycling between these forms basically, and one is the oxidized form and another is the reduced form, and we can have NH dependent other GR residues. So, thyroxine reductase will find, and what is happening therefore, that if you have the thiol residue on one part or the protein part, you will ending out with the disulfide bond. So, disulfide bond formation is important, and selenium can be there for your this glutathione peroxidase unit, that is the corresponding active site. Instead of S minus you can have the AC minus thing, and which can again it can form a disulfide bond like this disulfide bond and selenosulfide bond is important for all these activities. Sometimes we form the selenol, selenol can react with thiol. So, selenol, thiol, all these things are involved. So, that is why along with all sulfur bearing important molecules, selenium can take part in the biological world, because it can be incorporated in the corresponding amino acid. So, that is why it is very useful for our protection against oxidative stress, removing hydrogen peroxide and organic hydroperoxides. Is oxidized to selenic acid and the corresponding selenonate, that means corresponding alcohol function, then glutathione and glutathione reductase. There are altogether 25 selenoprotein genes in our body also. Humans, these are genes are there. So, once you know the protein, know the amino acid, you should also be able to identify, and that has been identified that you can have so many numbers of genes. Then lastly, we will see what is thioredoxin reductase, because it will be correlated to our next class, where we will talk about the corresponding chloride or iodide in the biology. So, how this is there? So, you can have the corresponding TRX, that means the thioredoxin and thioredoxin reductase. It can also show peroxidase activity by degrading H2O2 to water. So, it can also settle between these two forms, that means the reduced form and the oxidized form, and in the oxidized form you have the disulfide linkage, which we call as the S2, and the reduced forms are SH. Where we require this for cell growth, inhibited apoptosis, transcription factor, gene transcription, ribonucleotide reductase reaction, DNA synthesis, and finally the TRS peroxidase activity. So, not only knowing the TRS peroxidase activity, but we should also know about the different types of activities, what thioredoxin reductase can help us in doing all these reactions. So, this we will also know about when we talk about the corresponding things in all these enzymes. So, thioredoxin is the enzyme reduces the oxidized form of it, an electron donor for the ribonucleotide reductase that just now we have seen to give you the corresponding deoxyribonucleotide. So, it is another route basically ribonucleotide reductase is always we know for the DNA synthesis. So, we will have the electron transfer involving NADPH or FAD to FAD also, because you are having different E0 values for these two molecules. Then the link between cis and sec, that means selenyl sulfide bond, SS bond, instead of SS bond you have SAC bond, and that is useful for all these reactivities. So, like the disulfide bond, the SS bond formation between the cysteine residues, we can modify the structure of the peptide chain, and many such disulfide bonds are important for the assembly process of the protein, giving a particular type of structure. Selenium can function is a bimodal, two types, the bimodal role, bimodal biological action depending on the concentrations. At lower concentration it can show one type of reaction, at higher concentration it can show another type of reaction, that is called bimodal. It also finally the new findings people are working right now about whether it can show something or it can stimulate the anti-tumor immunity of the human body. So, references starting from your selenium page to then before that you can have the sulfur page for Wikipedia and the book of Crichton. Okay. Thank you very much for your kind attention.